So let's now talk about the endocrine pancreas. As I've mentioned previously, each gland is unique in its own way. Pancreas is a mixed gland, exocrine part 98% of which secretes the enzymes, and the endocrine part barely about 2%. But very important part, that endocrine part is synthesizing and secreting the insulin, and the insulin lack or resistance to insulin action results in diabetes mellitus, which is worldwide must be the number one killer disease, and therefore very important. This disease, diabetes mellitus, actually is included in all the subjects of medicine. Therefore, we will stick to the physiological aspects. Therefore, let's begin. Endocrine pancreas is basically only 2% total pancreatic mass, and there are cluster of specialized endocrine cells which are called as islets of Langerhans. These islets have three cell types. Let's see the location. The alpha cell, which is this cell in the outer rim of the islet. We are talking about the islets of Langerhans. And which cell is located in which part of that islet? Outer rim of the islet has the alpha cell. It constitutes about 10% of the total population, and it secretes glucagon. Beta cell 75 to 80%, center of the islet, and it synthesizes and secretes insulin. Delta cell only about 5%, and it's dispersed. On the outer rim is glucagon, central portion is the beta cell, but the delta cells are dispersed, mostly near the periphery of the islet. It secretes somatostatin. We have already described this somatostatin in the GIT in the context of acid inhibition. Then we discussed it as growth hormone inhibiting hormone, and now we will see here it's the remaining endocrine part. And then there are F cells, or also called PP cell. It's in the posterior portion of the head of the pancreas, and it releases pancreatic polypeptide. All right. Now, developmentally, the functional islets appear at the beginning of third trimester of the pregnancy, and the blood flow through the islets passes from beta cells to alpha and delta cells. Hence, the first cells affected by circulating insulin are the alpha cells. Do you get this point? The blood flow is such that it first goes to the beta cell and then to the alpha and delta cells. So the beta cell has synthesized and secreted insulin. It will then go to the alpha cell. Therefore, alpha cell will be the first cell to be affected or influenced or acted upon by the insulin. Insulin inhibits the glucagon secretion from the alpha cell. Therefore, let's talk about the insulin now. I already mentioned there is a tug of war going on between insulin on one side and all the other hormones on the other side. You have glucagon, glucocorticoids, growth hormone, all these G's, then catecholamines. They are all hyperglycemic and diabetogenic hormones. On the other side, there is this lone insulin, which is anti-diabetic hormone. Naturally, it's hypoglycemic. It reduces the blood glucose. Insulin, the anabolic hormone. Insulin is the primary anabolic hormone. It promotes glucose uptake and utilization by the muscle and adipose tissue. Now, remember forever that the two tissues which are the primary targets of insulin, muscle and fat, adipose tissue. It will promote the glycogen formation from glucose. Basically, we saw that the other hormones, they will cause glycogenolysis, and blood glucose will be level will increase. Insulin, on the other hand, It is an anabolic hormone. So it's going to store the glucose in the form of glycogen. Glycogen is a polymer of glucose, and therefore in that form the glucose will be stored. Anabolic hormone means protein synthesis will also be promoted by insulin. It promotes protein synthesis, it promotes TG synthesis, it will inhibit the lipolysis and protein degradation. Look, therefore the actions can be divided in two parts. One, anabolic action, the positive action, promotes glycogen storage from glucose, promotes protein synthesis, promotes TG synthesis, carbohydrates, proteins, fats, and prevents what? Prevents glycogen breakdown, prevents triglyceride breakdown, prevents protein degradation, right? And therefore, if there is a lack of insulin, then all these actions will be reversed, and that is what happens in the dreadful disease, the diabetes mellitus. Right, let's see the introduction, the synthesis, structure, secretion. Insulin is a protein hormone. We already are done with that part. It belongs to a gene family that also includes IGF-1, IGF-2 and relaxin. Relaxin we will see in the reproductive system. Synthesized as a precursor as pre-pro-insulin. That's also the case with all the other peptide or protein hormones. A large polypeptide precursor, then it will be cleaved to form the active hormone. Pre-pro-insulin will be the first to be formed. Then it will be cleaved to form proinsulin, and then proinsulin will be cleaved to form insulin and C-peptide in equimolar concentrations. That's important because look, the C-peptide is released along with insulin in equimolar concentrations. Therefore, sometimes to check the beta cell activity, we are talking of beta cells of the islets of Langerhans. To check the beta cell activity, sometimes they check the C-peptide levels in the plasma, because after all beta cell is forming this pre-proinsulin, proinsulin and then insulin and C-peptide. So levels of C-peptide will be indicative of how much beta cell is functional. Alright, connecting peptide or C-peptide. It has 31 amino acids and therefore it has some utility in the sense that you can check the C-peptide levels. Proinsulin has only 7 to 8% biological activity of insulin, but the C-peptide has no known biological activity. Now coming to the structure, insulin, one alpha chain, one beta chain, and the chains are connected by disulfide bridge. Insulin is stored in the secretory granules in the zinc bound crystals, and granules release, as I mentioned, equimolar amounts of insulin and C-peptide into the blood. Right. Half-life of insulin, 5 to 8 minutes. Look, it's among the shortest. We've already done with that part. Shortest half-life is that of catecholamines, a few seconds. Growth hormone, around about 2 minutes. Insulin, 5 to 8 minutes. Right. What is the primary stimulus for insulin secretion? It's the glucose. I want to introduce one term here, the substrate controlled hormone or substrate regulated hormone. Insulin is a substrate regulated hormone. What do we mean by this? Look, glucose is a substrate for insulin. Insulin is going to act on the glucose, and the glucose, the plasma glucose, is the one which regulates the insulin secretion. I mean, if there is hyperglycemia, insulin will be secreted, hypoglycemia, insulin will be suppressed. We will see how insulin is secreted in response to this substrate, its substrate, glucose. Here is a pancreatic beta cell of the islets of Langerhans, and glucose The plasma glucose has increased after we eat the carbohydrates, after we eat the food. Plasma glucose enters the pancreatic beta cell and it's metabolized. Once it's metabolized, there is ATP generation. And these ATPs are formed, they will act upon ATP sensitive potassium channel. The ATP sensitive potassium channel will be closed once these ATPs are generated. Now, potassium, which is expected to go out through its channels, because potassium has a concentration gradient from inside to outside. Now it cannot go out because the potassium channels are closed. The ATP sensitive potassium channel is closed. Therefore, the potassium just comes near the membrane. From the center of the cell, it will just come near the membrane. It accumulates inside the membrane. It had decided to go out. I mean, it is expected to go out, but it can't. Therefore, it just comes near the membrane and accumulates inside the membrane. This is the depolarization of the membrane. Obviously, inside of the membrane becoming positive, that's depolarization. This will be sensed by another channel, which is voltage gated calcium channel. The voltage gated calcium channel will open, calcium will enter the cell, and finally it will cause release of this insulin by exocytosis. Insulin will be released by the beta cell. This is the sequence of events in the insulin release, right? Therefore, we can think of it in the reverse, just for the sake of conceptual understanding. Look, insulin is going to be released by exocytosis. For exocytosis, what is required? Calcium is required. But calcium cannot be kept inside the cell, inside the cytosol. We have already described this in the general physio. The calcium, it cannot be kept in the intracellular fluid or in the cytosol. Cytosolic concentration has to be kept very, very low, else the cell will be damaged completely. Right. Therefore, it needs calcium, and calcium can come from the ECF. Calcium can come from the ECF after its channels are opened, which are voltage gated calcium channels. And therefore, the membrane voltage has to change. Only then this calcium channel will open. Membrane voltage is changed by who? By this potassium. Potassium comes near the membrane and accumulates inside the membrane. Why did it accumulate inside the membrane? Because it could not go out. Why it could not go out? It was supposed to go out, but then its channel is closed. Which channel? ATP sensitive potassium channel. I am deliberately going in the reverse to put the logic behind these events, why the sequence occurs in such manner. Right, that's ATP sensitive potassium channel. And who generated those ATPs inside the cell? The glucose, plasma glucose. It entered the cell and generated those ATPs. The drugs that target this particular protein, the ATP sensitive potassium channel, they will be called as insulin secretagogues. Sulfonylurea is a category of drugs. You will learn this in pharmacology. Sulfonylurea is the class of drugs which are called as insulin secretagogues. They cause insulin secretion. How? They have a receptor which is called as sulfonylurea receptor. which is located on this protein. Sulfonylurea will bind to that receptor, it will bind to this protein, and the next events will follow. That's the insulin secretion in response to its substrate, that's glucose. Insulin secretion is stimulated by hyperglycemia, obviously. That's the most important stimulus. Increased amino acids, increased free fatty acids, and also GIP, GLP. Remember, we have done this in the digestive tract. There are certain substances which are called as incretins, as in incremental insulin secretion. They are GIP, GLP-1, glucagon-like peptide 1. When we consume glucose orally, these are released from the GI mucosa and they go to the pancreas and they cause further insulin secretion. Look, oral glucose, so it's going to increase the plasma glucose already, and that increased plasma glucose will directly act on the pancreatic beta cell and will cause insulin secretion, which we saw just now in the diagram. In addition to that, incrementally, the oral glucose will cause release of GIP or GLP-1, and these GIP or GLP-1, they will go to the pancreas and will cause further additional insulin secretion. There are drugs which are being developed which will have this incretin effect, right? GIP, GLP-1, well, let me just complete this GIP thing. It was called as gastric inhibitory peptide, but now it was renamed later on as glucoinsulinotropic peptide. CCK, secretin, gastrin, epinephrine, they all cause release of insulin. What are the inhibitors of insulin? Hypoglycemia, I mean the reverse factors. If the blood glucose is low, then we don't want insulin to come in. Therefore, insulin will be suppressed. Decrease amino acids, decrease free fatty acids. So, first three factors you can remember, the three nutrients. If they increase in the plasma, they will stimulate insulin secretion. If they decrease in the plasma, they will inhibit the insulin secretion. Then somatostatin. Remember that delta cell in the pancreas? It is going to suppress insulin as well as glucagon secretion. Here we are talking about insulin. It's inhibited by somatostatin. Then leptin. We have described leptin in some other chapter. Leptin and epinephrine via alpha receptor. Note one point. Epinephrine acting via beta receptor stimulates the insulin secretion, and epinephrine acting via alpha receptor suppresses the insulin secretion, because then you will need glycogenolysis and increase the blood glucose. So, just see the dual effect of epinephrine via beta and alpha receptors, and parasympathetic stimulation as well. Right. Next, talking about the insulin receptor. It's called as receptor tyrosine kinase. If you remember the initial chapter, we have described that the receptors in the cell membrane are of two types. One are called as GPCRs, G protein coupled receptors, and the others are catalytic receptors. Means receptor itself has catalytic or enzyme activity. Insulin receptor is receptor tyrosine kinase, meaning that the receptor itself has the tyrosine kinase enzyme activity. The receptor present in the plasma membrane, the cell membrane, and it has got two subunits, alpha and beta. The alpha subunit is on the external aspect of the membrane and has got the hormone binding site. And the beta subunit spans the membrane and has tyrosine kinase on the cytoplasmic aspect of the membrane. Binding of insulin to the alpha subunit leads to autophosphorylation, and then beta subunits will cross phosphorylate each other on 3 tyrosine residues in the intracellular portion of the receptor, and then the phosphotyrosine residues then cause phosphorylation of tyrosine of a host of proteins. I mean, inside the cell, target cell, there are number of proteins, and those proteins have tyrosine residues. Now, insulin has come and it binds with the receptor, it binds with the alpha subunit, and then the beta subunit does the following things. There will be autophosphorylation. Tyrosine kinase activity has been activated in the receptor, and then it will act on the proteins, and it will act what part of proteins? On the tyrosine residues of those proteins inside the cell. What are those proteins? Insulin receptor substrates, IRS family, GAB1, SHC, P62-DOC, SIRP, that is signal regulatory protein. These are all the targets of the insulin action. Right, so that's how insulin acts. Next, glucose transport into, now insulin is going to act, and insulin going to act on the target cell. What is the mechanism of action of insulin? It causes glucose uptake by the cells, and this glucose uptake mediated by the insulin, it has got a transporter which is the GLUT4. We have seen this in general physio, so you can refer to that once again. There are GLUTs which cause facilitated diffusion of glucose, glucose entry into the cells. Insulin causes glucose entry into the muscle and in the fat cells by causing upregulation of GLUT4. So please remember, GLUT4 is insulin dependent glucose transporter. That's the action of insulin. It upregulates the GLUT4 synthesis, and thereby it translocates the GLUT4 in the membrane of the target cells in the muscle and adipose tissue, and that is how glucose will enter the muscle and the adipose tissue. Please remember, in the diabetes mellitus, when there is absolute or relative lack of insulin, or if there is insulin resistance, you will see that there is weight loss, and the weight loss is because there is loss of fat and there is loss of muscle glycogen and muscle tissue. Therefore, the person becomes thin. Right. How is it that the insulin receptor activity is terminated? Internalization. Once the insulin has initiated its action in the target cell, the receptor will be internalized into the cell, and dephosphorylation by the protein tyrosine phosphatase. There are tyrosine phosphatase inside the target cell. Once the insulin action has begun, then the receptor will be endocytosed, means internalized into the cell, and the tyrosine phosphatase will terminate the activity of insulin receptor. Right. Let's see the metabolic actions of insulin. We have so far described how is insulin secreted, what are the stimulators and inhibitors. Now, and the insulin receptor, the GLUT4. Now we will see the actions of insulin. So when we consume mixed meal, it will stimulate the insulin release from the beta cells. An important point to be understood is insulin then inhibits the glucagon release from the adjacent alpha cell. We saw this at the start of this section. There is beta cell, and when insulin is secreted by the beta cell into the blood, and then that blood goes to the alpha cell. That's how the blood flows. So insulin will go to the alpha cell, which is neighboring cell, and it will first stop the glucagon secretion from the alpha cell. Naturally, because now it's the insulin role after the food has been taken. Insulin is going to utilize those nutrients. It will make the body utilize and store those nutrients. Right. And therefore, insulin to glucagon ratio increases in the hepatic portal vein as it enters the liver. Insulin has come into the blood, it suppresses the glucagon secretion, and now it is circulating. Now when it comes to the liver or in the hepatic portal blood, insulin to glucagon ratio will be very, very high after food intake. Glucagon was suppressed, insulin was secreted. Okay, what is the action in the liver? It promotes glucose uptake and glucose utilization. It promotes conversion of glucose into storage form, that is glycogen. Well, this is now quite obvious. We have described it already at the start of the chapter. So, glycogenesis, and it prevents glycogenolysis. It promotes glycogenesis, glucose will be stored in the form of glycogen, and it prevents glycogenolysis, prevents the breakdown of glycogen. In the adipose tissue, it promotes the glucose uptake in the fat cells also by increasing the GLUT4 availability. GLUT4 is going to cause glucose entry into the fat cells. Action is lipogenesis and anti-lipolysis. We have already seen this, that synthesis of the nutrients, storage of the nutrients, and prevent the breakdown of those nutrients. Insulin stimulates the expression of lipoprotein lipase enzyme. Look, in the fat cell membrane, there is this, the capillaries which will have this lipoprotein lipase enzyme, and therefore increased fatty acid synthesis and formation of VLDLs. That's the action of insulin in the adipose tissue, in the fat cells. Fatty acids enter the adipocytes and there will be synthesis of triglycerides. Fat will be stored in the fat cells, and it decreases the lipolysis. Fat breakdown will be prevented. In skeletal muscle, yes, again the same. Insulin increases the glucose uptake by increasing the availability of GLUT4, the transport protein for glucose. Increases the glucose utilization and increases glycogenesis. Glycogen formation is promoted. It increases the amino acid uptake and protein synthesis. You must think like this: all three nutrients, carbohydrates, fats, proteins. And what is the action of insulin? Promotes glycogenesis, glucose converted to glycogen, and prevents glycogenolysis. 2. Protein promotes protein synthesis. Therefore, amino acid uptake is promoted by insulin and prevents the protein breakdown. And same for the fat, promotes the triglyceride synthesis in the adipose tissue and prevents lipolysis. Lipid breakdown is prevented. How does it achieve this, all these things? By increasing the expression of the enzymes. Increasing the expression of the enzymes. It will increase those enzymes which are involved in these processes. For example, glycogen synthase, which promotes glucose conversion to glycogen. Now, this will be upregulated by insulin so that glycogen formation is promoted. Right. One more important feature now. Note this point. I want your complete focus for now. Glucose is pushed into the cells by insulin. Along with that, potassium is also pushed into the cells. Plasma potassium will be pushed into the cells under the action of insulin. Please remember, glucose and potassium are co-transported almost simultaneously in the cells. Therefore, remember, in the hyperkalemia, you would give insulin, it will push the potassium from plasma into the cells. Right. Finally, let's talk about the diabetes mellitus. We have described all the actions of insulin, the mechanism of action, the receptor, the GLUTs. Let's quickly go through this. I am going to only stick to the relevant part in physiology. Diabetes mellitus, you know, it's a very important disease. It's a disease in which there is absolute or relative lack of insulin. Okay. Either it could be the absolute lack or relative lack, means insulin is there. It's not completely absent, but it's not enough to promote all those actions. Or there is insulin resistance. That's also possible. Insulin is there, but resistance to its action. Or it can be both. The most characteristic feature of DM, diabetes mellitus, is hyperglycemia. All the problems of diabetes mellitus are because of the hyperglycemia. First of all, let me just add here. We have seen in the diabetes insipidus. What is diabetes? The common word in diabetes mellitus and insipidus was diabetes. So, diabetes is running through. Therefore, the high urine flow, flowing through or running through. That's diabetes. What is mellitus? Mellitus is honey-like. It was discovered, this disease was known because urine which was increased in volume, there was loss of glucose in urine, and therefore honey-like, that's mellitus. Okay, most characteristic feature is hyperglycemia, and it may also result in glycosuria. Glucose will be lost into the urine. Type 1 and type 2. Type 1 diabetes has been called as previously called as juvenile onset diabetes. It starts in a very younger age, and it's called as insulin dependent diabetes mellitus, IDDM, because there is an absolute lack of insulin, and therefore the person has to be given administered insulin exogenously, right? It's insulin dependent. It starts at a very young age. Mostly this type 1 has autoimmune nature. There are antibodies that will destroy the pancreatic beta cells. And the classic triad of symptoms, you know that. This is a general knowledge. Polyuria, polydipsia, polyphagia. Excessive urinary output, excessive thirst, and excessive hunger. Since glucose is there in the plasma but is not able to enter the cells are hypoglycemic basically, and that if the hypothalamic neurons, they are not able to take up the glucose and metabolize it, there will be constant hunger, polyphagia. Right, hyperglycemia leads to increased glucose, blood glucose, and then this glucose is filtered by the the kidney. When it starts flowing in the renal tubular fluids, it will pull water by osmosis into the renal tubules, and therefore urine output increases. Therefore, this is called as osmotic diuresis. Please remember this term. Diabetes also exhibits osmotic diuresis, increased urinary output. And polyuria, because of this osmotic diuresis. Then because fluids have been lost, there is excessive thirst, and as I mentioned, it's a catabolic state, it's a breakdown state because there is no insulin. Therefore, weight loss and muscle wasting. Yes, no protein synthesis because there is no protein anabolic action of insulin. Muscle wasting. Absolute lack of insulin results in diabetic ketoacidosis. We have slightly described it previously. Ketone body formation. Look, the beta oxidation of fatty acids and the metabolism of fats requires some amount of glucose inside the cells. In the insulin deficiency, there would not be this glucose available inside the cells, and therefore incomplete oxidation of fats results in the formation of ketone bodies. That's diabetic ketoacidosis. It results from the abnormal fatty acid metabolism. And they are acidic. Therefore, it will be a metabolic acidosis, diabetic ketoacidosis. Okay, type 2 diabetes, it's NIDDM, non-insulin dependent diabetes mellitus. What does it mean? It is due to the insulin resistance. I mean, insulin is there, but there is a resistance to its action in the target tissues, and therefore particularly seen in the obese individuals. The obese individuals are likely to suffer in later life uh, with type 2 diabetes mellitus. There is a possibility of insulin resistance getting developed